Hello, Bismillah Rahman Rahim. My name is Dr. Mohammed Azhar. I am representing the topic of my presentation is 3D genome regulation. My name is Dr. Mohammed Azhar, Assistant Professor, PhD from USSC, China, postdoc from Sussex, China. Served in Comstas University as a lecturer assistant professor for more than 10 years, and now I am serving as a assistant professor in Chorasan University of Veterinary Sciences, Bahawalpur. So today is our topic is 3D genome regulation, which is highly unknown topic in our country, especially, and which is one of the latest and hot topic in the worldwide in the researchers and the genomics, especially era for people who are researching on genomic specialization on genomics. 3D genome regulation is kind of a hot and novel topic, and this explains how the genome is which we know today usually in Pakistan and the normal molecular biologist, according to molecular biologists, the genome is 3D, is not 3D, which is linear. But the recent studies of 3D genome regulation from last 5, 10 years, 5 years especially, have led the discovery to discover that 3D genome, the genome is not only linear, it is highly active, and the 3D space or in the nucleus, and it acts spatially and temporally at specific time and space tissue according to the need of the body, cell, organism, condition, stress, and other response to external stimuli or internal need of the body. So in that kind, the genome transcription is not always the complete transcription of all the genome. It is selective. So when it is selective, so gene is the similar kind of this. Every cell in the body have the same genome, but they have the different transcriptome and the proteome according to the situation and the expression, and that is regulated by the 3D genome regulation, which is also known as the epigenomics and genomics, because it is regulated at the chromatin level. When the chromatin level, the suppression marks disassociate, it will let the opening of heterochromatin into euchromatin, and which will in the end allow the interaction of cis-acting regulatory elements like enhancer and promoter at distant spaces to interact and allow transcription of certain genes. This 3D arrangement influence the transcription program, especially in stem cells, as we can know here. What are stem cells? You know it better. Stem cells have the capacity to regrow a fully functional organism. Role in the transcription regulation. Enhancer promoter roles, which are cis-regulatory. Enhancer promoters are distantly placed regulatory elements in the higher eukaryote, especially in human, and their physical interaction is necessary for the transcription activation. So that is done through the looping mechanism, which has explained here. But 3D genome regulation explains the better of it. 3D interactions are cell type specific and dynamic. Misregulation of these structures can lead to disease or development. There are a certain level of genomes. If we if we see the nucleus under microscope at a certain condition, there are the chromosomes, and not all the chromosomes are active, and not the every part of the each chromosome is active. So there are selectively chromosomes which are active, and the selective parts of certain chromosomes are active. So chromosome, those are divided into different categories. Which are active categories, we divide them into active category, and which are non-active, which we divide them into inactive categories. So chromosomal territories. So according to their activation, we divide into chromosomal territories. Like individual chromosomes occupy distinct nucleosomes. For example, A B compartment chromosome, gene-rich compartment, transcriptionally active, often central. B compartment, gene-poor rich regions, which are not actively transcribed. Topology associated domains. There are topology tagged, which are highly active domains which are inactive parts. So in A and B figure, we define what are active and B inactive parts, and then the TADs, which are transcription activation domain. Cis regulatory element, as we know that there are different kinds of cis regulatory elements, especially enhancer and promoter. Earlier we have discussed that how they interact by using the loop mechanism, and now we can discuss here the regulatory element direct linear specific, which are enhancer loop, and how they do it by looping mechanism. Here we explain it that how they interact because they came close to each other physically with the help of certain transcription factors. They interact with each other. Mammalian genome contains roughly about 2,000 tags covering more than 90% of the mappable genome, and varying in the genomes are 100 kilobases, 1 MB each apart, distant. So genome-wide between loci, there are on average separated about these tags are 150 kb, including approximately 30,000 interactions, active interactions between putative enhancers and promoter regions. So how there is an example how they are active and inactive, and how they change the TADs and active parts of the chromosome and inactive parts during the lifespan, or the you can say the spatio-temporal space and time, the growth time, or the disease response, how they interact, how they respond to it by activating or by inactivating certain genes. So here we present an example. Pluripotent cells, as you have known, is a kind of a type. Embryonic stem cells have a more open and dynamic chromatin because they are at that time of growth, they are expressing more and more proteins. They are expressing also their specialization, specializing certain proteins. So at that time the most number of the expression of proteins is higher. The then we came to the embryonic stem cell, how frequent long range interaction between active genes occur. We can study from here because this is the most active you can say the phase of the life. Upon differentiation, after differentiation, specialization, TAD and gamma become more defined because now the specialization have occurred. Now the tags will be a kind of a more defined and leading to lineage specific expression. Embryonic stem cells, however, derive from the inner cell of mass. What are embryonic? Here we explained that blastocyst, embryoblastocyst, and the induced pluripotent are somatic cells programmed to become pluripotent cells because they are the normal somatic cells which are which when introduce four basic transcription factors like Yamanaka factors, they become pluripotent cells. Yamanaka got the Nobel Prize for this work. 3D genome reorganization during differentiation is different. Like different differentiation involves the switching of compartment, activation and activation. The certain genes are first in the embryonic stem cells, as we see the most of the genes are expressing. Then the specialization occurs. Then the selective genes are active and inactive according to the different organelle and space and time. So this is the how it explained. The enhancer promoter contacts are weird to support new transcriptional programs. The architecture becomes more stable and aggressive in lineage committed cells. And however, when we study this example here, it is a 3D genome level for organization. There are, there are two types of cells, embryonic cells and fibroblast cells. As you can see, in fibroblast cells, embryonic cells, because most of the cells are active, so there is a tad, this picture and the heat map in both sides, A and B compartment, showing that the TADs are active in A and B compartment in embryonic stem cells, which means most of the genes are active. But on the later stage of development, whereas fibroblast, the TADs are different. It's the same TAD which is active TAD1 in A compartment becomes highly inactive in compartment B, as shown in the figure. So this explains that the how the TADs genes are active and genes are transcribed, and how the physical interaction is more and more in the earlier development phases, which is embryonic stem cells, which is the one of the earliest and highest replicating and transcriptionally active phase of the life. So in fibroblasts, it becomes less active. So CTCF is one of the certain transcription major transcription factors involved in the gene regulation, and it is also called as the master behavior of the genome. Zinc finger protein that marks TAD boundaries, usually found at the TAD boundary. Cohesion protein complex that helps form and maintain loops. So mediation and complex transcription help bring enhancer close to the target promoters. Implication was stem cell biology, 3D genome architecture is the key to maintaining pluripotency and enabling the plasticity. So pluripotency is established by the 3D genome regulation, and selective gene regulation is also, plasticity is also maintained 3D genome activity, is which controls the gene activation at different time frames of development. Disruption in the 3D structure can lead to loss of pluripotency or the improper differentiation. So, if there is any problem in the differentiation, they can play a role in the loss of pluripotency of the cells. Structure 3D genome recognition gives insight into the reprogramming and development. So if we studying 3D, so if, if we study the 3D genome, we can see the how the genes are actively transcribed during the different phases of life, and how we can use, or we will be able to understand that how the reprogramming of the cells and or in the development, it plays a significant or major role even before the gene regulation. The epigenomics or the 3D genome regulation plays a significant role. So there's we have there are some after the post-genomic era, there are so many kind of omics techniques have been developed experimentally, and then bioinformatics to do downstream analysis to study the 3D genome regulation. The one of them is the chromosome conformation factor. This is the basic technique, and there are several techniques. 3C, 4C, 5C, high C is mostly used. Chia loop is also used. Chia pet is also used. They have different functionalities, like they can, it depends upon the locus. One versus one means one locus versus one locus. One locus means one against all locus. Many against means many against, and all versus all is high C is used for in the 3D genome regulation. If you want to target the specific transcription factor and study the locus, then G and loop is also studied. The GFF is also studied. So these are some of the significant studies which are used to study the gene regulation. This is the high C protocol, general protocol, the cross-linking, cutting with the enzyme, field ends with the biomarker, and mark with the biotin ligase and purifying DNAs, sequencing and pair use. And then we have the conclusion 3D genome regulation is fundamental regulatory of a gene expression, spatial architecture, determined via compartment adds and enhancer promoter loop mechanism is basically the fundamental mechanism. Topological features are dynamic during the development, which we are explaining and have explained in this presentation, that in the earlier embryonic stem cells, and the later stages of the developments are like fibroblasts, they have the topological features like the tags or the transcriptionally active domains on the basis of the 3D genome. They change with the passage of the time in the development and disease regulation also. Disruption in 3D structure contribute to disease. Also, aberrant tag boundary and also hijacking or loop can lead to misreading or oncogene or dermal. 3D genome plasticity underlines the cell-fate transition, and they can reproduce. Technological advances are unlocking 3D genome flow. Certain advancements in the high different experimental technology like high C, single cell, and 3D genomic super resolution mapping imaging, now allowing for the common structure and unprecedented resolution, more resolution depth, and we can study the interactions, paving the way for precision gene regulation and regenerative medicine.